All right. So thank you so much for the invitation to come to this symposium. It's really exciting. I mean, Claudia's talk was amazing, and I'm sure the rest of the talks will be as well. I'm very excited to be here, despite the fact that I had to miss the first Warriors playoff home game last night, but they won anyway, so I'm happy. All right, so my speaker is on. That's not good. All right, so I actually have a different title. Decided to talk about something a little different today, and so I'm going to talk about dances with nuclear condensates and the interplay between three different condensates, heterochromatin nucleoli, and a new one that we've discovered called Sheila. All right, so Claudia gave an amazing introduction to nuclear architecture. I'll just remind you about the focus here, and that is that nuclei have numerous bodies, each of which have distinct components, distinct functions, and distinct structures. And so the two most prominent ones are heterochromatin and nucleoli, and those will be two of the things that I'm going to focus on today. They generally constitute about 2/3 of the volume of the nucleus, but there are many, many other bodies. Other chromatin states, such as polycomb, are associated with bodies. Different functions, such as DNA repair, replication, transcription, telomeres, splicing, et cetera, are all associated with bodies that have distinct components that are associated with the functions that they encode. Now, looking at these types of pictures makes me wonder whether there really is such a thing as nucleoplasm, and whether the nucleus is really just filled with these distinct bodies. Now, most, if not all, so all of these are membraneless. The only membrane in this picture is the nuclear membrane. All of them are membraneless, and most, if not all of them, behave as liquid-like, what we like to call biocondensates. And Claudia gave you an introduction to phase separation. Likely that these are formed by phase separation or similar mechanisms. Now, I recognize that using the term phase separation is somewhat controversial at this point. And I think that it's good to hark back to Schrödinger, who in his book, What is Life, where he basically said, well, biology follows the natural laws or the laws of physics. And I'm going to paraphrase here. But there's a lot of shit loaded on top of it that obscures the fundamental physical laws. And I think that that is one of the issues here. We can see phase separation happening, whether it's liquid-liquid, liquid-gel, whatever, in vitro, very simple systems. It's much harder to prove in vivo because you can't generate the same kind of phase diagrams that you can do with purified components in vitro. However, you can see all the signs of liquidity, molecular mobility, et cetera, et cetera. I think more importantly that the question is not whether phase separation happens in vivo or not, but we have all these bodies. They behave in many cases as liquid-like entities. How are they formed? How are they maintained? How are they dissolved? How do they impact genome and nuclear organization and function? To me, those are the most important questions. These are entities that we need to understand, and that's what we're trying to do, along with many others in the field. Now, we spend a lot of time working on the heterochromatin. I'll give you a little bit of a flavor for that. But what we've started to think about is how do these different condensates interact both structurally and functionally? So what is the interplay at both the structural and functional level between these condensates? And that's what I'm going to talk about today. Very much work in progress, but I hope this will generate some discussion and some thinking on your part as well as mine. All right, so our favorite condensate is the pericentromeric heterochromatin. And I want to make a distinction, which is that the term heterochromatin is often used now, especially in the genomics field, to refer to any silenced locus in the genome. And that's not what we're talking about here. Although there are commonalities, what we're talking about here is the fact that, and here's the Drosophila karyotype, but the same is true for mammals, including us, and that is that Around the centromeres, the region where the kinetochore forms, it's responsible for attachment to the spindle and chromosome segregation, there are megabases of highly repeated sequences, so-called satellite DNAs, simple sequences like AAGAG, repeated for hundreds of kilobases, up to megabases, and also transposable element fragments. These surround the centromere on all chromosomes. So there's very large domains, and that's distinct, and also at the telomeres. That's distinct from much smaller regions of silent chromatin, which might have the same composition in terms of histone modifications and reader proteins and things like that. But those are much smaller regions. Okay. Now, even though the pericentromeric genomic regions are separated in terms of being on different chromosomes, if you look at the interphase nucleus, they coalesce to form what are called chromocenters. And so the organization in the nucleus reflects their ability to come together in three-dimensional space and form clusters, as we saw in. All right, so pericentromeric heterochromatin, or PCH, is what I'll be calling it, are liquid-like condensates that are associated with repeated DNAs. There are few genes in the pericentromeric heterochromatin, but there are in fact genes there. In Drosophila, for example, the only copy of MAP kinase and the only copy of the Rad21 cohesin cleasin are actually embedded in the so-called silent region, and these are essential genes. So there are genes there, but for the most part, what the pericentromeric heterochromatin does is mediate chromosome segregation, things like meiotic pairing, obviously centromere function, sister cohesion, and silencing of genes and transposable elements. We, along with Gita Narlakar's lab a few years ago, I guess 6 years ago, showed that the pericentromeric heterochromatin behaves as a liquid-like condensate. And I'm not going to go through all of the evidence that we presented then. Excuse me. But the bottom line is that we spent a lot of our effort looking in vivo. Gita spent a lot of her effort looking in vitro. And so both in vitro and in vitro, we could see all the properties of liquid-liquid phase separation reflected in this condensate. Now, the mechanism, the molecular mechanism, is that these repeated sequences are highly enriched for one particular histone modification, histone H3, lysine 9, di and trimethylation. And its associated reader protein, epigenetic reader protein, a heterochromatin protein 1, or HP1, which contains a pocket called the chromodomain that specifically and uniquely binds to di or trimethylated histone H3 lysine 9. Now, I'm not going to go into the details. We have a lot of work that we've done focused on how these form in vivo. And basically, yes, you have recruitment of HP1 to K9 methylated chromatin through this chromodomain. But in reality, it's this giant hairball of interactions. That is, HP1 exists predominantly as a dimer, and it interacts not only with itself and with the chromatin, but it also oligomerizes, so that oligomers of dimers. It also interacts with DNA or RNA through disordered domains. It, at the dimerization cleft, has the ability to bind hundreds of different binding partners, each of which are themselves multivalent and contain disordered domains and other structured domains that mediate weak hydrophobic or electrostatic interactions. So it's this giant hairball of interactions held together by weak multivalent interactions. All right, so I did want to stop before going, or pause, not stop, before going into today's topic and try to at least give you some sense of why I'm excited about thinking about the role of condensates in chromatin biology. And that is because it raises new possibilities for thinking about regulation, and for both in terms of organization and things like gene activation or DNA replication and things like that. And I think this is really important in the context of thinking about engineering, not just genome engineering, but how we engineer the nucleus, that we need to be thinking about something beyond how genome regulation may involve open versus closed chromatin. This is really pervasive in the field, that either you have open chromatin or closed chromatin. I think Claudia showed very nicely how there's much more complexity there with the addition of the polymer. But I think, if we think about it, liquidity can have major impacts on genome organization, such as the ability to form hubs or compartments from self-fusion. So when you have condensates of like composition, they can undergo liquid-like fusions. If they don't have the same composition, they won't fuse. HP1 condensates will fuse with each other. HP1 condensates will not fuse with polycomb condensates because they don't have a molecular compatibility. And so, for example, we had shown a few years ago, Grace Lee, who was at the time a postdoc in the lab, that if you look at transposable elements strewn throughout the euchromatin, they are actually small regions of K9 methylation associated with HP1, but they, in 3D space by high C and by FISH, actually associate with the pericentromeric heterochromatin in this manner. Another possibility, which we're currently testing, is that instead of thinking about the spreading of chromatin states, which is a well-known property of the heterochromatin, as a processive enzymatic reaction, which people like Jerry Krabte have essentially ruled out based on the kinetics, is that maybe spreading of chromatin states is more of a wetting process, again, reflecting. The other thing is that instead of thinking about chromatin compaction, that is the inability of things like Pol2 to actually enter into a region because the chromatin itself, the DNA nucleosome polymer, is compacted. Once you start to invoke condensates, you can think about the biophysical properties of the condensate as a whole, such as the ability to selectively exclude or retain genome regulators independent of the packing of the chromatin fiber underneath that. And then finally, from work that we did on DNA repair, where we see that double-strand breaks made inside the heterochromatin domain. So this is HP1 shown here in red, and these are repair foci arising after irradiation. We showed that, in fact, these foci have the double-strand breaks have to move outside the heterochromatin domain and actually go to the nuclear periphery to complete homologous recombination repair. And although Jolla and others have shown that once they leave the heterochromatin domain, they move to the periphery due to actin-myosin mediated directed movements. That is not the case in terms of how they leave the domain. And so one of the things that we've explored is the idea that, and I think Andy has actually worked in this area, and that is this behaves as that the DNA nucleosome polymer is behaving as a copolymer with multiple different epigenetic states all along it, and that you can really understand how different parts of the polymer can move in and out simply based on the energetics of the ability of, for example, if you are K9 methylated, you will interact with HP1. And if, as we see in all these different, we've described this in different publications, during repair, the initial stage of repair, K9 methylation is removed, that part of the polymer can no longer interact favorably, interact in terms of the energetics with the heterochromatin domain and will essentially ooze its way out. So again, much of this is fantasy. We're testing a lot of these ideas. In fact, if we go in and directly demethylate a region of the heterochromatin, we can actually see it moving out of the heterochromatin domain. But these are all works in progress. But I think what I'm hoping is that you get an idea that it's much more than just potentially much more than open and closed chromatin, and that we need to come to grips with that. Okay, so this is the theme today. I'm going to talk about, hopefully, interplay among three condensates if I don't run out of time. So first, we have the pericentromeric heterochromatin that's shown in pink and nucleoli shown in green. And then we have a new condensate, which we call Shelob, and you'll see why, that actually is present in all the interchromatin spaces outside in the euchromatic space, but also surrounds the heterochromatin, the pericentromeric heterochromatin. So there's like this three-layered structure. And so we want to understand how is this organization established, and are the associations important for organization functions? And again, this is all new, unpublished, works in progress. So this is, let's first focus on nucleoli and the pericentromeric heterochromatin. This is a highly conserved organization. You see it in human nuclei. So here's the heterochromatin surrounding nucleoli. You see it in Drosophila, plants, et cetera, et cetera. The work I'm going to talk about in Drosophila was done by a really talented postdoc, Barsha Rashikar. And so I told you about what the pericentromeric heterochromatin is. Nucleoli, as I'm sure many of you are familiar with, is a multi-layered condensate, as first shown by Cliff Brangwyn when he was a postdoc in Tony Hyman's lab. And the key to understanding nucleoli is that they're built on another repeated genomic element, the ribosomal DNA. These are much larger repeats, depending on the organism. In humans, there are 45 kilobase repeats, but there's hundreds of copies located on different chromosomes. But the key to understanding nucleolar formation is that it's transcribing ribosomal DNA. So for the formation of ribosomal RNA from RNA using RNA pol 1, that actually creates the initial layer, and this is called the fibrillar center. And then there's a second layer that's formed by the recruitment of processing factors, things that will process the ribosomal RNA, such as one that I'll focus on, which is fibrillarin. And then in the outer layer, you have what's called the granular component, which is where ribosomes are being assembled. And each of these, like in the case of all the different condensates within the nucleus, really reflect different condensates or different phases. They have different composition, different sets of molecular interactions. So this is the site of ribosome production. Now, in flies, in humans, our DNA is embedded in the pericentromeric heterochromatin. And so you might imagine that this association is simply because of genomic proximity. But it turns out, so here's flies, there's about 200 repeats each of the rDNA on the X and the Y, but in fact, none on the second or third or fourth chromosomes. And yet, they're also surrounding the nucleolus. Okay, so it's not simply because you are you have a physical association in cis that you have this surrounded configuration. All right, so what Varsha did was to look at the initial establishment of nucleolar and pericentromeric heterochromatin condensates in early Drosophila embryos. This is really a wonderful system because you can look at the very first stages of formation of these bodies and not worry about whether you're just looking at a maintenance stage as in cultured cells. So early Drosophila embryos, you have fertilization, and you have a bunch of nuclear cycles. You don't get cellularization until cycle 14, and then you have gastrulation and you make a larva. What's interesting about these cycles is that they're just SM cycles, or DNA replication, mitosis, DNA replication, mitosis. And these interphases, especially early on, are incredibly short. They're 6 minutes to start with, 15 minutes in cycle 13, and 90 minutes in the first more extended interphase in cycle 14. Now, heterochromatin foci, as Amy Strum in my lab first appear in cycle 11. You need to reach work that I'm not going to talk about. You have to achieve a certain concentration of K9 methylation on the chromatin. That requires this many cycles. That's when heterochromatin foci first appear. Nucleolar foci, as shown in the Wieshouse lab, first appear in cycle 14, so 2 cycles later. So 13 is the first time when you see both. And so what we're going to be looking at is a bunch of movies where we're marking the heterochromatin with HP1, RFP, and the nucleolus with fibrillarin GFP. Now, when they start out in cycle 13, they are not in this surrounded configuration. And by later in development, they are. And so the question is, how do you get from here to here? And so one important point to make is that in every single one of these mitoses, both condensates dissolve. All condensates that we know of dissolve in mitosis. So in every interphase, they have to redo this process of starting out with all the molecules being diffuse, then nucleating, and then growing. Now, in these early cycles, as you'll see here in cycle 13, there's only 15 minutes in interphase, and so they don't have a lot of time to grow, and they don't undergo fusions. And so this is what you're going to see. I guess there's a little too much light, but you can see that they come out of the previous mitosis, they're diffuse, and then heterochromatin bodies first appear, then nucleolar bodies appear because ribosomal RNA is starting to transcribe. But they remain separated in space, and there's not enough time for them to grow and undergo liquid-like fusions. In the next cycle, in cycle 14, you see something very different. They start out exactly the same as they did in cycle 13, but they now have 90 minutes to grow and fuse. And so that's what you're going to see here. They're now growing much larger, the green, the nucleolar components, HP1 and fuchsia. And you can see them, again, creating much larger, here's a much larger volume, occupying much larger volume of the nucleus. This is the side view. Bottom line is that in at this stage, by the time you get to the end of cycle 14, you're still not in this surrounded configuration, this canonical, highly conserved configuration. You have what is a more elongated branch structure for HP1 juxtaposed with the nuclear periphery, the nuclear lamina. And then there's one part of the heterochromatin, which corresponds to the sex chromosomes, the rDNA locus, which is in fact in sort of inverted, that's surrounded by the nuclear material. But the important thing is that if you now look at subsequent cycles, I'm not going to dwell on this, I want to get to the functional studies, is that what you see is that in cycle 15 and 16, you're still in this elongated configuration. In cycle 16, it starts flipping back and forth between this, the association with the nuclear lamina and the surrounded configuration. And it's only in cycle 17 that you now get a stable surrounded configuration. Okay. So the bottom line here is that this canonical organization is assembled by a very dynamic process. It just doesn't appear de novo. There needs to be a bunch of stage-specific intermediates to get from this point to this point. I didn't get into it, but there's a lot of interesting, I think, physics, biophysics to think about because of changes in nuclear volume and changes in the speed with which this kind of like with which these different structural changes occur. Becomes shorter and shorter and shorter in each cycle. So we don't understand that, but we're trying to figure that out. All right, mechanistically, what could be happening? Tons of things we don't understand. So it could be changes, as I said, relative volumes of the two condensates, aspects of the nuclear volume changing, wetting of two immiscible liquids, et cetera, et cetera. Lots of different possibilities we're investigating with different types of experiments. We've also, and I'll come back to this, but just as a hint, we've been working with Sam Safran's group at the Weizmann Institute, integrating physics and different types of simulations. And this has led us to this idea of, to favor this idea of this being formed through some kind of affinity hierarchy, just solely based on interactions between the different molecules. Before I do that, I want to turn to the question of, is this, this has been descriptive. Here's what the dynamics are. I really wanted to talk about this, not because that's particularly interesting, but because I think it's really important to keep these dynamics in your head and understand what's happening live. But first, I wanted to turn to whether manipulating one condensate or the other impacts the ability manipulating one of the condensates impacts the organization of the other. And I'm not going to go into details of when we get rid of the heterochromatin, the nucleoli become bigger, you get much more ribosomal RNA. We think that this is not an interaction between condensates per se, it's because most of the rDNA is silent, and it's silenced by K9 methylation in HP1. So if you get rid of K9 methylation globally and you reduce the amount of heterochromatin, you get a much bigger nucleolus simply because it's transcribing more rRNA. It's possible there is interaction there. What was more interesting came out of looking at what happens when you get rid of the rDNA. And this was done by the Wieschaus lab back in 2017 using a what's called a compound chromosome, a highly rearranged chromosome in Drosophila, something you can do with Drosophila genetics. And basically you can create embryos that have no ribosomal DNA at all. Now these animals live till late embryos simply because the mother sticks in tons of ribosomes into the egg, okay? But the important point is that all these stages that we're looking at when you normally form the heterochromatin and the nucleoli, they're perfectly normal in terms of timing and all that sort of stuff. So they're living high and they're living well off mom's ribosomes. All right, so what happens when you get rid of nucleoli? And there's a number of really interesting phenotypes, and they raise a lot of important points and questions. The bottom line is, here's plus rDNA, you have the nucleolus, you have the heterochromatin. This is cycle 14, it doesn't yet surround the nucleolus. Here's what happens in the minus rDNA case. First and foremost, fibrillarin, this nuclear component, has nowhere to nucleate. There's no rDNA, there's no rRNA. So it actually goes off because it has inherent self-association and forms what we call a neocondensate. It forms this other beautiful spherical object, but not connected to the heterochromatin. And that's what you see quantitated here. other nuclear components, like this outer nuclear component module, it just gets dispersed throughout the nucleus. And that's because it doesn't have self-association. You'll see there's a third pattern, abnormal pattern that happens. All right, so that's one phenotype. The second phenotype that we were very excited about is that, in fact, the pericentromeric heterochromatin becomes abnormal. And it becomes hypercondensed. You don't see this elongated configuration you see in wild type. It's much more condensed. You can measure this by looking at the aspect ratio. And so you can see in the minus R DNA case, it's significantly more condensed than in the plus R DNA case. And if you do fish with different satellite sequences, you can see in wild type, they're much more dispersed throughout this volume, and they're much more compacted in the absence of the RDNA. So what we conclude from this is that nucleoli actually are required to regulate the distribution of the pericentromeric heterochromatin in 3D space, and especially to keep the heterochromatin from hypercompaction. We can discuss later about, is that because of an inherent property of the polymer that's K9 methylated plus the HP1, but there is this ability to become more compacted if you don't have this nucleolus to assemble around. All right. There was another phenotype that really turned out to change how we thought about this process in fundamental ways, and that was this. In subsequent cycles, in cycle 17, which happens to correspond to the cycle when you get this surrounding configuration, what we saw was the formation of not this, within this compacted pericentromeric heterochromatin, so this is the minus R DNA case, we see this mysterious void appearing, a black hole or whatever. And you can see this in this movie. So here's the fibrillarin neocondensate, and you can see down here and up here, you can see as this cycle 17 moves forward, this void appears. And you know, for a while it was like, oh, that's sort of interesting. But what's really interesting are these neocondensates that form. That's now this persists throughout embryogenesis. You see this hole here. And so, yeah, what's in there? So Varsha went and looked, you know, all sorts of candidates. It's not RNA. There's no DNA in there that we can find, or at least not high concentrations, no histones, no nuclear components, a whole list of things that aren't there. What is there is protein, and this is just showing a pan-protein label using the 488 NHS ester. Here's the void that's formed, and then here's a high concentration of protein. It looks a lot like what happens in the post-RDNA, where there's one of the highest concentrations of proteins in the nucleus is in the nucleolus, but that's not a nucleolus. The fibrillarin, remember, is somewhere off to the side. All right, so what are these proteins, and what can they tell us about nuclear heterochromatin assembly? So this is where our collaboration with the Saffron Lab really paid off. And so in working with Omar and Gaurav and Sam, we were thinking about, okay, what kind of proteins could be involved in both assembling the heterochromatin around the nucleolus and in creating this void in the abnormal case where you lack the RDNA. And so we thought about it and came up with this idea that, well, maybe they're amphiphilic proteins, of course, your standard protein X. And the properties that it should have is that it needs to self-associate, and most importantly, have affinity for both the nucleolus and the pericentromeric heterochromatin. Okay. Pretty simple, but it took us a while to get there. And so the idea is that this protein X would be able to sort of glue or stabilize the association between the pericentromeric heterochromatin and the pericentromeric heterochromatin in the nucleolus. And in the case where you have no RDNA, due to its self-association and its affinity for HP1 pericentromeric heterochromatin, would form or fill this void. And you know, this is would be another type of neocondensate, right? It's an abnormal structure that forms because you've lost your primary nucleation site. And now proteins that are used to undergoing weak interactions with other proteins or nucleic acids will now go off and find another place to accumulate. And so with Sam, Omar, and Gaurav, well, they did this. They developed a 4 minimal component coarse grain model to help us think about this. And it's very simple, and I'm not going to go into great detail here, don't have the time. But basically, we have the heterochromatin as a DNA nucleosome polymer. Embedded in that is the rDNA, our RNA. Remember, this is all coarse grain. Fibrillary as just a representative of the nucleolus, and then this protein X, this amphiphilic protein shown here in yellow. And the bottom line is that we set up an affinity hierarchy. And we decided that this was based on known properties. Others were guesses. Bottom line is that we tested a range of affinities. We didn't just tune this so we got to get the result that we wanted to get. I'm not going to go into detail for this, but basically this type of hierarchy fits what we know about these proteins and about these condensates. And so if you run the simulations, you end up being able to recapitulate the surrounded configuration, again, over a range of affinities, not just by tuning it to one or the other. And I think most importantly, we were able to also show that using the same modeling, that if you remove the rDNA, and therefore you get rid of these affinities, okay, the ability of rDNA to interact with fibrillary as a nuclear component, fibrillarin's interaction with this magical protein X, then in fact you can recapitulate again over a range of affinities the Fib neocondensates and this void that is filled with this protein X. Okay, all this is well and good. That just tells you what's possible. It doesn't tell you what happens. And so, is there a protein X? And if so, what is it? And we got very lucky. And I'll tell you why we started out picking this protein. We got lucky and identified this protein pitchun, which is a dead box RNA helicase. And the reason that we picked this protein, which not a lot is known about in flies, there is a human homologue called DDX18. Number one, this protein has nucleolar affinity. Okay, it sits in the right place. It's in the outer layer of the nucleolus. It's not just at the surface. It fills the whole outer layer of the nucleolus. And so you can see that here. Here's fibrillary in pink. So that's a more central component. And then this is this pitchun component around the edge. So it fulfilled one of our criteria. It has affinity for the nucleolus. Second is that it also has affinity for the pericentromeric heterochromatin. It contains what's called the PXVXL motif, which is a motif that is necessary for interacting with the dimerization cleft of HP1. So most of the hundreds of HP1 binding partners contain this PXVXL motif. Okay, and then secondly, from the work from the Wieshouse lab, they didn't look at HP1, but they noticed that in the minus rDNA embryos, pitchun accumulated in this body, an abnormal body, which looked a lot like Barsha's images of what the HP1 looks like in the minus rDNA embryos. Okay, that was suggestive, but certainly not proof. And the bottom line is that yes, obviously I wouldn't be talking about this if this wasn't true, but it turns out that yes, pitchun is not only located in the outer part of the nucleolus, juxtaposed with HP1, but in the in wild type, but in the minus rDNA case, it fills this void. Okay, so this is the HP1 here, and this is this pitchun protein tagged here. So you can see that in all these different embryonic nuclei, that's true. And I think you can see this in movies. And so you can see this is again the cycle 17. It starts out where the HP1 is in this condensed state. And then as the cycle proceeds, pitchun gets more and more and more pitchun gets recruited. And we think this is generating this void. Okay. So together with the affinities, it suggests, obviously does not prove, that pitchun is at least behaving like this protein X. And we think that it is likely mediating the layering of the nucleolus and the pericentromeric heterochromatin in wild type, and the formation of this void in the minus rDNA case. And so finally, can we show that if you get rid of pitchun, you actually lose this surrounded configuration? And the answer is a tentative yes. We did an RNAi experiment in S2 cells. Unfortunately, if you knock out, to get rid of pitchun in embryos, you have to first get rid of it in oogenesis, At least that's what we tried to do, and it doesn't make eggs, so you don't get any embryos. So it's actually essential for oogenesis. We'll get around that. So we did this in cultured cells, and in fact, what you see is what we would have predicted, which is that instead of this surrounded configuration, you have a much more compacted HP1. It surrounds less of the nucleolus, and that's exactly what we would have predicted. Now, from this, we tentatively conclude Pichun is required for the surrounded configuration. And the tentative part of this is this data from last week, and we need to do a lot more quantitation, and we have a lot more experiments to do in general. How much time do I have?
Good.
I know I'm good, but how much time do I have?
20 minutes.
20 minutes?
You want, we're ahead of schedule.
Well, let me start a whole different talk. No. All right, so then I have time for the last 10 minutes. There we go. All right, so from this part, the first 2/3, the conclusions are, number one, the formation of this surrounded configuration, this interaction between different condensates is highly dynamic. And then we also, from getting rid of the rDNA, conclude that nucleoli are in some way regulating the distribution of the pericentromeric heterochromatin and prevents its hypercompaction. We think that based on the modeling as well as in vivo experiments, that there's a hierarchy of interaction strengths that underlie this surrounded configuration. And in particular, we've identified one protein. There may be others. We're looking for those, doing proximity labeling. But it seems that at least Pichun is at least a linker between the nucleolus and the pericentromeric heterochromatin and fills this void in the minus rDNA. And I want to, I'm going to emphasize this again at the end, but I think one of the most important lessons, and maybe the one thing that I would ask you to remember from this, is that I think that what we're seeing with respect to manipulating these condensates individually and collectively is that there are enormous implications for mutant phenotypes that we need to consider. And this is based on seeing these neocondensates and mysterious black holes. If you lose the nucleation site for a condensate, remember it's based, the whole formation of these, at least liquid-like condensates, is based on weak multivalent interactions. If you lose the nucleation site or get rid of it, or you get rid of one domain in a multivalent protein that's responsible for interacting with other proteins and recruiting them into the condensate, then you need to keep in mind that the things that it's going to interact with, such as what we saw with fibrillary, you get rid of the rDNA, fibrillary goes off and does whatever it wants to do. In this case, it self-associates, but other proteins will go and associate with secondary sites or tertiary sites or quaternary sites. And so I think that's really important. And we've seen in this case, so we have 3 different phenotypes associated with getting rid of the rDNA. Some proteins just get dispersed. Those could be neomorphic in terms of their effect on phenotype and disease. Some make neocondensates like fibrillary, they just go off and self-associate. And then the third was this Pichun behavior, which is that it now fills this void simply because it has both self-association and the ability to interact with HP1. All right. Now, the last part, I'm going to talk about this other condensate that surrounds the pericentromeric heterochromatin. And there are themes that are now coming together, and specifically this hierarchy of affinities. But as you'll see, we haven't quite put it all together yet. This is work from a former graduate student, Joel Swenson, and a staff scientist, Serafin Colmenares. So we got into this protein because we identified it in an IP mass spec experiment using HP1 as bait. So it's an HP1 interactor based on that. We, at the same time, are doing an genome-wide RNAi screen looking for things that disrupted HP1 organization in the nucleus. And it was the only protein, like the Highlander, it was the only protein that came out of both screens. All right, I know that the Highlander reference is not, maybe 2 people here get that. Yeah, okay. All right, so AlphaFold is pretty useless for this. And the reason for that is that this is a 969 amino acid, or 919 amino acid protein. It has 3 structured domains, zinc fingers. So there they are in the AlphaFold, and the rest of it is just flopping around in the wind. Bunch of disordered domains. It is a pain in the ass protein to work with. Do not ask me why we haven't done in vitro phase separation. experiments because you can't get the damn thing out of bacteria. Anyway, it has all these disordered domains as shown here, only these 3 zinc fingers. So it is a bona fide HP1 binding partner. In reciprocal IPs, if you pull down HP1 or you pull down Shelob, you will pull down the reciprocal. And like with Pachun, it contains one of these PXVXL domains here down in the C terminus. So it looks like your standard, biochemically identified binding partner, right? And most importantly, it's required for organismal viability. It's also required for forming the PCH condensate. So if you get rid of Shelob, this is in mutants or by RNAi, then you lose the HP1 condensate. So here it is in, sorry, that's very small, but in heterozygotes versus homozygous mutants, you also lose pericentromeric heterochromatin functions. You lose the ability to silence genes. This is a LAC-Z integrant that gets silenced. And then if you can alleviate silencing using what's called the Suvar mutation, that is mutating something required for heterochromatin formation, Shelob gives you also an increase in gene expression. It also alters if you mutate it, alters this, or it's required in wild type for the spatial temporal dynamics of DNA repair in heterochromatin. So it's impacts pericentromeric heterochromatin organizationally or structurally and functionally. But there's no change by ChIP-seq in HP1 genomic localization or protein levels if you do live studies or Western blots or IF. So it's very weird. Mostly that's a change. Those are changes you would see with something that affects the organization and function of heterochromatin. Even weirder, it's very different in its distribution. So most HP1 interactors, which we published back in 2016, either completely co-localize with HP1, here's HP1, here's an interacting protein, or they form these little subdomains inside the HP1 condensate, but not Shelob. By confocal imaging, what you see is that it has 2 patterns. It's not exclusively surrounding the heterochromatin. It has a web-like pattern that fills the interchromatid spaces. So sorry, here's Shelob. So out here, it's filling interchromatid space. I'm not showing you, if you stain for histone, they're mutually exclusive. So it's in the interchromatid spaces. That's why we named it Shelob, by the way, for the giant spider in the Lord of the Rings. So it's the web-like pattern, but it also has this enrichment, and it's rich in region very low histone density, and it's enriched around the pericentromeric heterochromatin. So Shelob is in red, and so you can see it doesn't fully surround it, and there's some other localizations, but it does show enrichments in parts of the pericentromeric heterochromatin. But it doesn't actually physically overlap, and so we collaborated with Kachu and did super resolution imaging. This is the scan over here. And basically what you see is that there's just a few points of contact between Shelob, and that's what the white represents, just a few points of contact between Shelob, shown here in green, sorry for keep switching the colors, and HP1 in fuchsia. All right, now, Shelob actually behaves like an independent condensate, that it exhibits liquid-like properties. If you look by FRAP, it's highly mobile, just like HP1, not like H2A. But I think if you look live, it's really a lot of fun, because what you can see are all these liquid-like properties. You can see nucleation, growth, and fusions going from M to G1. And what you see as you go from G2 to M is some hypercondensation and dispersal. And in fact, there's this really amazing pattern. Sorry, I won't go too deeply into this, but remember I told you that in mitosis, HP1 and nucleoli dissolve, okay? So what happens with the Shelob protein is it's surrounding the HP1 and has this web-like pattern in the euchromatin. And when HP1 dissolves, so you'll see here, all of a sudden, all the Shelob rounds up. And so anyway, that's just liquid-like behavior. It then dissolves a little later in prophase. So we think that Shelob is an independent liquid-like condensate. And so it's a very strange protein. It fills the interchromatin spaces in the euchromatin, excludes chromatin, behaves like a liquid-like condensate, and it infrequently contacts the PCH condensate. And yet, it's required for pericentromeric heterochromatin organization and function. So this is not behaving like a typical biochemically defined interacting protein. What we think is happening, and this is based on doing analyses of HP1 mobility in using different types of methods, FCS, FRAP, hexanediol treatment. The bottom line is that if you knock down SHELOB, then you see a significant increase in HP1 mobility in both the euchromatin and the heterochromatin. And if you overexpress it, you see a significant decrease in mobility of HP1. We've done flip experiments. You also can show that in the absence of SHELOB, you get more HP1 diffusion from inside to outside, the HP1 node. So this suggests, we think that the answer is that SHELOB regulates HP1 mobility and diffusion, and that's how it affects the organization and function of the PCH. So again, these are ideas, but basically that maybe SHELOB behaves as a shield that helps recycle HP1 into the domain, or it somehow recruits a stabilizing factor. But the bottom line is it's not behaving like a typical binding partner where you would say, oh, it's impacting it simply because it's binding it throughout. And in this case, it's acting in a sense at a distance, but there is this connection. All right, so just to end, how does this bring us back to the question about how layering occurs, how different condensates interact? And we have not modeled this yet. We're working with Sam now to try to bring this into the same framework, coarse-grained framework that we did for the nucleolus. But the idea is very similar. The idea here is that SHELOB has self-association and HP1 has self-association, but that the ability to interact, and these are weak interactions, but I'm saying this is stronger than the ability for these molecules to interact with each other. Okay, so this is a weaker interaction. And so the idea is that stronger homotypic interactions, that is homotypic with respect to each condensate, so HP1, SHELOB, SHELOB, is what drives the formation of separate condensates as they emerge from this diffuse pattern into this separated pattern, so demixing. But that it's the weaker heterotypic interaction that is between SHELOB and HP1 that drives the actual layering, the association of those condensates together. So very similar to, think of this as pitchoun and HP1. In this case, now it's HP1 and SHELOB on the outside, but it's the same principle. And so, in fact, we have shown, I'm not going to go into it, we have shown that the PXVXL on SHELOB is in fact a weaker PXVXL compared to other HP1 interacting proteins. And that's, we suspect, based on structure, that's also true for pitchoun. And so this predicts that altering the concentrations or affinities will disrupt the condensates and the layering. We've started to do that. If you overexpress SHELOB, so you now increase this interaction by increasing dosage, you actually get mixing of HP1 and SHELOB. And we can show that that's dependent on this PXVXL. If you mutate the central V, then you in fact no longer get mixing even with overexpression. And then conversely, we've started to play with, okay, if we decrease the strength of the self or homotypic interaction, in this case by decreasing HP1 binding to K9 methyl, then in fact you can again now see mixing for enrichments of SHELOB out in the euchromatic region. So bottom line is we think that if we now bring these together, that this layering SHELOB, HP1, the nucleolus is mediated by affinity hierarchies, specifically amphiphilic proteins with weak heterotypic affinity, contrasting that with stronger stronger homotypic affinities. And the important point here is you don't need to have a protein that just sits at the surface of one condensate to allow it to be interacting with the other. These are proteins that are actually distributed through hundreds of microns away, but it's just that at the surface, because they're held together in their own condensate, they can at the surface interact weakly, and that's what we think is keeping them together. And again, I think we need to think about when you have these types of interactions, the implications for genetic studies, et cetera, et cetera. All right, with that, thank you for your attention. Thanks to quote team, well, it is really a team now, but it's mostly in my lab, it's just Varsha. Really talented postdoc, but we've had a really great collaboration with Sam and Omar and Gaurav. And then the Shelob work that I talked about was predominantly Sarapin. And thank you. Thank you to our funders as well. And sorry to the rest of the lab.